To organize the millions of species scattered across Earth, we need a system built on precise, non-arbitrary rules. It is often assumed that growth and reproduction define what is alive. However, non-living crystals can accumulate mass and grow, while many biological organisms, like mules or sterile worker bees, do not reproduce at all. The actual, non-negotiable requirements for life are three specific properties: a constant internal metabolism, a cellular structure, and sensing the environment. Applying these exact parameters immediately filters out non-living matter. It takes the chaotic swarm of the natural world and snaps it into a rigid, analyzable matrix. Without these strict parameters, biological classification would be guesswork. By enforcing them, scientists can construct a framework capable of mapping the entire biosphere. That framework relies on a taxonomic hierarchy. Designed by Carolus Linnaeus, it moves step by step from species up through genus, family, order, class, phylum, and kingdom. Moving up this ladder captures higher levels of structural diversity, culminating at the kingdom level. To organize life at that macro level, biologists rely on R.H. Whitaker's five-kingdom system, which sorts all organisms into Monera, Protista, fungi, plantae, and animalia. Whitaker sorted these kingdoms using precise biological criteria: the complexity of the cell structure, the physical organization of the body, the primary mode of nutrition, and known phylogenetic or evolutionary relationships. In 1990, Carl Woese added an even broader layer to this architecture. His three-domain system sorts all life into archaea, bacteria, and eukarya by analyzing deep genetic sequences. These ranks act as precise coordinates. When you identify an organism's place in the matrix, you are reading its exact location on the map of evolutionary history. Within the eukaryotic domain, a single shift in cellular architecture completely dictates an organism's ecological role. We see this clearly at the dividing line between fungi and plants. Kingdom fungi lacks chlorophyll. Built with rigid chitin walls and a branching mycelium network, they survive by decomposing organic matter. In contrast, plant cell walls are constructed from cellulose and packed with chloroplasts. The presence of chloroplasts gives plants the machinery to. This defines kingdom Plantae as autotrophs, anchoring them as the biosphere's primary producers. At the foundational base sit algae, categorized by specific photosynthetic pigments: chlorophyll in green algae, fucoxanthin in brown, and phycoerythrin in red. A microscopic cellular variance, chitin versus cellulose, dictates whether an entire kingdom operates as the planet's producers or its decomposers. Plant taxonomy serves as a historical record of environmental adaptation, mapping the migration of flora from aquatic environments onto dry land. We trace this migration through alternation of generations, a continuous loop between haploid and diploid phases. In primitive organisms like bryophytes, the haploid gametophyte dominates. As plants adapted to land, the diploid sporophyte took dominance, marking a leap in complexity. With that shift came structural upgrades. Pteridophytes, like ferns, developed dedicated vascular tissues, xylem and phloem, allowing for true stems and leaves. The final adaptation was the seed habit, found in gymnosperms and angiosperms. Encasing the embryo inside a protective shell bypassed the need for environmental water, securing terrestrial dominance. Higher plant classification tracks this exact progression, a clear, step-by-step journey from fragile, water-bound organisms to robust terrestrial seed-bearers. In the animal kingdom, invertebrate classification relies heavily on a single physical property: embryonic geometry. The most primitive baseline is phylum Porifera, the sponges. They operate entirely at a cellular level of organization and lack body symmetry entirely. Acquiring organized body symmetry is a major evolutionary filter. Coelenterates develop radial symmetry, where any vertical plane divides them identically. More complex animals develop bilateral symmetry. Only a single plane divides the body into equal left and right halves. After determining symmetry, taxonomy looks at the next developmental filter: germ layers. Early organisms are diploblastic, built from just two embryonic cell layers, ectoderm and endoderm. More advanced organisms are triploblastic, having evolved a third internal layer known as the mesoderm. Developing a bilateral geometry and a dedicated mesoderm layer provided the necessary biological scaffolding to evolve specialized, complex organ systems. Once the mesoderm is present, how that middle layer structures itself dictates internal complexity. The defining feature is the coelom, a body cavity lined by mesoderm. Acoelomates lack this entirely. Pseudocoelomates have a partial cavity. True coelomates possess a fully lined cavity, the mechanical prerequisite to house advanced cardiovascular and digestive architectures. Building on this architecture, the highest taxonomic macro level is phylum Chordata, defined by three structures: a notochord, a dorsal hollow nerve cord, and paired pharyngeal gill slits. Whether growing into a flatworm or a mammal, an animal's anatomical destiny is locked into place during the very first rounds of embryonic cellular division. The taxonomic blueprints map out cellular life with mathematical rigidity, but there are entities that break the system entirely. Viruses, and infectious RNA known as viroids, are biological anomalies. If you try to place them into Whittaker's five-kingdom matrix, the parameters categorically reject them. That's because a virus lacks cellular organization entirely. It is essentially an inert nucleoprotein crystal, composed of nothing more than genetic material encased in a protein shell called a capsid. Biologists have historically struggled to categorize them. When Louis Pasteur first studied these infectious agents, he simply gave them the term virus, the Latin word for poisonous fluid. The paradox is that outside a host cell, viruses are as inert as salt crystals, but the moment they breach a living cell, they hijack the host's machinery to replicate their own genetic code. Viruses exist in a permanent gray zone. They carry specific genetic instructions, yet they lack the independent metabolism required to be classified as alive. Even with these boundary exceptions, the biological classification matrix remains an exceptionally powerful predictive tool. Identifying traits like radial symmetry or triploblastic germ layers allows a biologist to plot an organism's precise phylogenetic coordinates. By simply reading an organism's assigned taxonomic rank, a scientist can instantly deduce its cellular architecture, trace its embryonic history, and map its evolutionary lineage. Biological classification acts as a functional schematic, an evolving matrix that engineers logic and order out of 4 billion years of chaotic evolution.